Hey everyone, welcome back to another episode on genetics. Today what we're going to go over is something called complementary gene interaction. And usually on exams, the way how this question is given to you as a complementation table, something like these, and then you have to solve them to figure out what their complementation groups are, what their genotypes are, and if you cross different individuals together, what effect will that have on the offspring? Like what phenotypes would you get? But to kind of go over complementation, a lot of people struggle on understanding how this particularly works. And complementation is basically another form of an epistatic interaction. So all this is, it's basically a coupled epistatic interaction. How we talked about before, where we have a certain recessive phenotype, and you can have different recessive phenotypes. And essentially, what we're trying to see is how many genes are contributing to a recessive phenotype into a dominant phenotype, or this is also referred to as the wild type phenotype. So basically all this epistatic interaction is that you have multiple different genes, and all genes have to be activated in order for you to have a dominant wild type phenotype. So in other words, let's say that in our example here we only have two genes. Both genes have to be dominant and present in order for this reaction to occur, in order for you to get a dominant wild type phenotype. So another thing is you can have multiple different genes as well. You can have 5 million genes, and basically if one of these genes are inactivated, like let's say this one is little b, both of these alleles have a null mutation and they do not contribute to a functional copy of the B enzyme. And then if you don't have the B enzyme, you cannot process this reaction to occur. Same thing is that if you had little a, this reaction will not be able to occur if you have little a, right? And if both of them are recessive, you still have a recessive phenotype. So yes, so basically all this reaction is that basically if you're big A, big B, whatever, you'll be wild type. If you're big A, little b, you're going to be recessive. If you're little a, big B, whatever, you're going to be recessive. And then little a, little b, you're going to be recessive. So basically, that's all complementation is. That is literally all it is, okay? Now, with this reaction, all this is just a simple, a product. You have, you have a product, but most of the time when you're getting asked this question on an exam, you're getting asked as something like this, where you have a table of 3 different individuals and you cross them together and you get something like this. So how do we solve that? To begin with, what you need to remember with complementation is that when 2 individuals are crossed together, so when 2 individuals are crossed together, there are 2 possibilities that you can have. The first possibility is that you have complementation. So basically, whenever you have complementation, that means that you're getting a dominant wild type phenotype from the cross from this following cross. So you're getting a dominant phenotype in the offspring. So you're getting that dominant phenotype in the offspring. Another thing to note about complementation is this is typically referred to as a plus symbol. I should actually rewrite that better. So this is typically referred to as a plus symbol in a typical complementation table, but it can be referred to as a different letter or something like that, which we will go over later. And whenever you have this plus symbol or this complementation symbol or whatever, if you're seeing complementation, that means that these two individuals, these original two parents must be recessive at two different genes. So basically, if you see that plus, what's happening is that when you're crossing these original parents together and you're seeing a wild type phenotype in the offspring, that must mean that those two parents had to have been recessive at two different genes, okay? Now, the other possibility that you have is that you have no complementation. And basically what this is that you're getting a recessive phenotype in the offspring. And this is typically referred to as a minus. And then what happens here is that if you have no complementation between two different parents, then therefore they must be recessive at the same gene, okay? So they must be recessive at the same gene. So as you can tell, right here, what we're trying to do with these tables is we're trying to figure out how many different genes at minimum are involved for this reaction to occur. Because we can have almost an infinite number of alleles for a particular trait, or even for a particular gene. So what we're trying to do is we're trying to figure out how many different genes are involved in this epistatic interaction, okay? So that's all complementation is, well, complementation tables are. So let's try out a simple complementation table over here. And so first of all, what you want to look for is what I prefer doing is I prefer going column by column. Some people also like doing it row by row too. Whatever it is, you can whatever you prefer the most, you're allowed to do whatever you want. I just prefer column because that's essentially how I've always been doing it. Okay. With these complementation table, essentially what you have here is right here what we have is individual 1 and individual 1 is crossed together. Whenever you have whenever you have two of the same individuals being crossed together, this information is kind of just useless, you know, because obviously they have to be recessive at a different locus, right? Or they obviously have to be recessive at the same locus, right? So that information is kind of useless. So you do not really need that information. But what you want to do is you want to start with individual number 1, and keep in mind these are all recessive individuals. So we're going to start with recessive number 1, and we want to look for what is his interaction with the other individual. So number 1 complements with both 2 and 3. So number 1 complements with 2 and 3. And then if we go over here on number 2 does not complement with number 3. So based on this information, if we know that number 2 does not complement with number 3, which is a minus, they must be recessive at the exact same gene, okay? So the way how typically these problems go is the first section of the problem is asking you to write down the complementation groups. So the complementation groups are basically correlated to the individuals that are recessive at the exact same gene. So this is basically equal to, your complementation groups are equal to individuals that are recessive at the same gene. So basically, because 2 and 3 do not complement with one another, therefore they must be in the same complementation group. Okay? So group 1 would be 2 and 3. And then we're left off with individual number 1. So what do we do with individual number 1? So individual number 1, we said that it complements with both 2 and 3. Because individual number 1 complements with 2 and 3, therefore number 1 has to be recessive at another different gene than 2 and 3. So therefore they would be in a different group. So this would be one will be in group 2. So that's essentially how to answer this question. So next, what you want to do is you want to look at what are the genotypes of each individual. So for the genotypes of each individual, for example, for group number 1, because we said that if 2 and 3 are crossed together and they do not complement with one another, therefore they must be recessive at the same gene. So what you can do with these problems, and these problems are basically pretty much arbitrary in terms of like what genotypes you use, but you can essentially use like any kind of gene designation that you want. So let's say for group number 1, for 2 and 3, they're recessive at the same gene. So let's say they're recessive at a, right? And then group number 2, because we know that group number 2 over here, when 1 is crossed with number 2, we're getting complementation. So therefore, they must be recessive at a different gene. So because number 1 has complementation with 2 and 3, they must be recessive at a completely different gene. So therefore, their recessiveness will be at gene B. So there will be little b. Okay? And then what we want to do now is, because we know that all these individuals on our table are completely recessive, right? Their genotypes cannot be all homozygous dominant for every single locus, because then they would be a dominant or wild type phenotype, okay? So what we have here is essentially we have two different genes that we figured out. So what you can do from here is that basically the number of complementation groups is equal to the number of genes. Okay? Okay, so now what we need to do from here is we need to sort of fill in the blanks, because we know we have two different genes, little a and gene A and gene B. What we need to do is we need to fill in the blanks. So let's say that all these individuals in our table are true breeding, so they can either be little a, or big A, and also little b, big B. So what we need to do is we need to cross these individuals together and determine what their genotypes are. So for group 1, for example, 2 and 3, let's say their genotype is little a, big B. If this is the case, and if you cross these individuals together, what we want to do is that we want to make sure that no matter what happens here, that you always get complementation. Because we know that if you cross these together, we should always see a dominant phenotype, okay? So let's say group number 2 has big A here as well. So what happens if you cross big A with little a? You always end up with a phenotype proportion that is all big A, right? Same thing if you cross big B with little b. What happens is that all your offspring are big B, little b. So they all end up having the dominant phenotype. So this is the correct genotypes of each individual. Okay, so that's basically all you have to do for these problems. Do not make it harder than it has to be honest, and this is what a lot of people struggle on. Okay, so let's do a little bit more of a complicated one. we only had a couple of, we only had 3 individuals there, but let's do one with 6 individuals, okay? And you can pause this if you want to try it on your own. So essentially, we're going to start off with individual number 1, which is over here. Individual number 1 does not complement with 3 and 5. So therefore, because they do not complement with one another, they belong in the same group, 1, 3, and 5. And because they belong in the same group, they must be recessive at the same gene. Okay? So let's call that gene that they're recessive at is little a. Cool. Now we've done group number 1. Let's start off with group number 2. Group number 2 is recessive at 2 crossed with 4, you get a recessive phenotype. So group number 2 is 2 and 4. So because now we have a different group, we have a different gene, a different locus that we're recessive at. So little b. So hopefully that made sense. We have number 3 over here. Number 3 is basically saying over here that number 3 is when you cross with number 5, you're getting a recessive phenotype. That is expected because, again, 3 and 5 are in the same group. Therefore, they're recessive at the same gene. And if you cross them together, and if they're recessive at the same gene, you're never going to get a wild-type phenotype, right? It's like group 1 over here. Let's say both of 3 and 5 have little a. So this is number 3, little a. This is number 5, little a. When you cross these together, you're always going to get a recessive phenotype, because as long as because in this epistatic interaction, you need all genes to be dominant in order for the wild type phenotype to be shown. Okay, be present. So basically, 3 and 5 are in the same group, which we've already confirmed. Number 4 is complements to number 5 and number 6, which we've expected it to be because number 4 is in the same group as number 2. Number 5 is complementing with number 6, and we've already established that number 5 is in the same complementation group as as group 1. Okay, and then that leaves off number 6. Number 6 complements with everything else, so number 6 will be in its own complementation group. So therefore, group number 3 would be recessive at a different locus, little c. So now all you need to do is fill in the blanks, okay? So because we have 3 different groups, we have 3 different genes, and because of this, typically the way how you would write this down is you would write it down as something like this: big B, little big C, and then group number 2 would be big A, big C, and then group number 3 would be big A, big B. So what you want to do here is you want to confirm that you have everything correct, okay? So what you want to do is you want to cross we want to cross number 1 together with number 2, which they're in a different group. So what we're crossing is little a, big B, cross with big A, little b. So if you cross these together, you always end up with a dominant phenotype because all of these genes are dominant. So this will be a dominant phenotype. So what we want to do is we want to confirm that's the case. So number 1 and number 2 should complement together, which they do over here. Okay, and you can do that. You can do that exercise, so on and so forth. Same thing if you want to cross number 1 with number 3. Then you end up with little a. You get a recessive phenotype, and you do not show any complementation. So remember, remember at the beginning how I've mentioned that as long as you have one locus that is completely recessive, homozygous recessive, you would always be recessive. Okay? So basically, you need every single gene to be either heterozygous or homozygous dominant in order for it to be a dominant or wild type phenotype. Okay? And the reason why you don't write these as heterozygous over here is because what we're assuming over here from the original table, we're assuming that each of these individuals is true breeding, and because they're true breeding, you would not write down heterozygosity here, okay? And that'll make your table a lot more complicated than it has to be. So you would always assume that they're homozygous for either for recessiveness or for dominant. And yes, so basically what you want to do here, like I said from originally, for determining their genotype, basically what will happen is most of the time with these groups, you would have 3 different groups and they would only be recessive at one gene. There are some problems which that might not be the case, but again, you would just have to use your critical thinking skills for those problems, okay? So let's do another problem where essentially we have to make a complementation table. Now this one is pretty easy. What you want to do is you want to combine the individuals in the same group. So remember, if they're recessive at the same gene, they must be in the same group. So these guys are in the same group. So when you cross 1 and 3 together, you should see a minus. And when you cross 2 and 4 together, you should see a minus. But then any other combination of crosses, you will see pluses, okay? So let's make that table. Okay, so to make this table, we have 2 different groups. So what we want to do is 1 and 3 do not complement. So we're going to write down that as a minus. And then everything would be plus. 2 and 4 do not complement one another. And that'll be a plus over here. 3 does not complement with 1, but we don't have a possibility to write down 1 over here. So we're gonna have complementation there. And that'll be it. That's basically all you have to do. Okay? Okay, and then last one, we're going to try another complicated one where now what we're trying to do is we've kind of figured out a way how to make the complementation groups and how to figure out their genotypes. But now what we want to do is we want to figure out if you cross certain individuals together, what will their phenotypes be, okay? So yeah, what you might have is something like a color one instead of just numbers. So we have a wild type phenotype that is gray, and we have all these other recessive phenotypes. So we're going to do the exact same thing. We're going to look for the ones that do not complement one another. So blue does not complement with black and pink. So group number 1 is blue, black, and pink. Group number 2 is white and red. Okay. Next one is black and pink, which we've already written down over here. So we're good with that. Pink we've already covered. Red we've covered with white, and then that just leaves orange. So basically we have 3 different genes, and they would be recessive at different genes. So group number 1, they must be recessive at the same gene. So group number 1, their genotype will be little a, oops, big B, big C, big C. Group number 2 will be big A, little b, big C, big C. And then group number 3 will be big A, big B, little c, right? So basically, we've kind of already had practice with this. But now the question is asking you, if you took a true breeding individual that is blue, and you cross it with a true breeding individual that is pink, how many offspring should be gray out of 54? So remember, their genotypes, and they belong in the same group, are little a, big B, big C, crossed again, little a, big B, big C. You end up with a phenotype proportion that is all little a, big B, big C. So none of them, so 0 individuals out of 54 will be gray. Because gray is a wild type phenotype, and if we have this one recessive, homozygous recessive locus, then we're going to be a recessive phenotype. Okay? So same thing over here. For this problem, you have a true breeding blue individual, and you're crossing it with a true breeding red individual. So how many offspring should be gray? So what we have here, blue and red are in a different group. So blue is what we wrote down as little a, big B, big C, crossed with a big A, little b, big C, red individual. So this is blue, this is red. So if you cross these together, the offspring proportion will be big A, big B, big C. So because we all have a dominant phenotype because we have a dominant allele at every single locus, we would have a dominant phenotype or a wild type phenotype. So 54 individuals will be gray. Okay, so let's do a little bit of another practice problem. Let's say instead now, let's say that this orange individual was essentially did not complement with any of them, so they'll be minus. So let's say we have a different situation like that. So how will that change our answer? So what we would have if individual orange does not complement with every single group, what happens now is that because they do not complement with every single group, they will be a part of every single group, right? So group number 1 will be blue, black, pink, and orange. And then group number 2 will be white, red, and orange. So what if that was the case and you have this situation, now you have 2 different groups. Their genotypes will be, we can write that down. Group number 1 will be little a, big B. Group number 2 will be big A, little b. And then because orange is a part of 2 different groups, we're going to write down a separate genotype for that one. Orange is going to be little a, little b. Because what we want to do is we want to make sure that they're recessive at little a. Because when you cross little a with little a, you're getting a recessive phenotype. And you're not getting any complementation. But then when you cross group number 2 with orange, you want to make sure that you're also not getting any complementation, because these recessive B alleles from both of these individuals give you a recessive phenotype. So that'll be another case that you might see on an exam. And just keep in mind that individuals aren't perfect in nature. They aren't, they're not going to be recessive and dominant at every single locus, and that is something that you have to take into account. But you can take that into account by just inferring on the complementation table and what you're getting, okay? So let's do another practice problem revolving around this information that we have originally. So group 1, group 2, and group 3. So let's say I tell you to write down a genotype of an individual that complements with group number 2, does not complement with group number 3, and half complements with group number 1. So for these problems, what you want to do is you want to take it one gene at a time. We know that group number 2 is the primary gene that we're looking at is little b here. So if the problem states that if an individual does not complements with number 2, therefore their gene would have to, or their gene for B would have to be big B, right? Because we're getting a dominant phenotype. And then it tells you that it does not complement with number 3. The primary focus on group number 3 is little c. So if you're not complementing with little c, you would have to be little c, right? And then it tells you that you have a half complementation situation with group number 1. And we're looking primarily at little a. So remember, what we've so far been doing is basically these crosses right here, either big A with little a, or little a with little a. But what's stopping us from crossing big A with little a? So if we cross these together, what we end up with is a situation where we have half of your offspring have the dominant wild-type phenotype, and then half will be recessive. So this is essentially what it means by half complementation. So if this individual half complements with number 1, they would be big A, little a. So now what you want to do is you want to make sure that every single one of these crosses actually works out and it does what it's supposed to be doing, okay? So what we want to do is we want to make sure that this individual does not complement with number 3, okay? So if we cross this individual together, what we end up with is that because both little c and little c are the same, and you cross these together, you'll have little c, therefore you're getting a recessive phenotype. And that criteria works out. And let's try it out with number 2 as well. If you try to cross number 2 together, basically the big C with little c, you're getting a dominant allele in the offspring, and then little b with big B, you're getting a dominant allele in the offspring. So that's basically that. And then half complementation, what we've covered over here, it's basically that. So hopefully this helped out. I know it's a pretty it's the longest video I've done, but a lot of people struggle on that. Don't try to make complementation harder than it has to be. But yeah, that's it.